In this presentation, I'll be reviewing the central dogma and the mechanism of transcription, pre-mRNA processing, export, translation, and various mechanisms of mRNA decay or degradation. I'll finish by then reviewing microRNA and the current models of gene silencing by the RISC complex. My intended audience will be graduate students, meaning this lecture will be fairly advanced, and I'll be moving fairly quickly. If you have any questions or notice an error, please comment and let me know. And thanks for watching. So we're going to start with the transcription initiation of mRNA. In order to begin producing mRNA, we need our RNA polymerase II holoenzyme to bind the promoter of our gene. In general, there are about 4 or 5 different elements at work here. We have enhancers, which are far upstream of our gene, or even on a different chromosome, and they function to recruit transcription factors that promote the recruitment of RNA polymerase. We have a core promoter element like a Tata box. The Tata box is found at about 40% of gene promoters, and it's just a short stretch of DNA rich in thymine and adenine that recruits Tata binding proteins found on the RNA polymerase holoenzyme. Genes lacking a Tata sequence will often have a downstream promoter element actually found downstream of the transcriptional start site, and the downstream promoter element actually recruits RNA polymerase II by binding to TF2D, which we'll discuss in a second. Another alternative to the Tata box is the initiator motif. The initiator motif is a 17 base pair element that also binds directly to TF2D. In addition to the enhancer that binds transcription factors and the downstream promoter element, initiator motif, in the Tata box that bind to RNA polymerase II, we have nucleosomes or histones. These are inaccessible regions of DNA that must be removed by histone remodeling proteins like the Saga complex and many others in order for the RNA polymerase to function properly. So generally speaking, promoters contain enhancers that bind transcription factors, core promoter elements like the Tata box or the initiator motif that bind RNA polymerase II subunits, and histone remodeling proteins that pave the way for transcription. RNA polymerase II contains at least 12 different proteins, each of which has their own subunits. And one of the most important proteins in RNA polymerase II is the TF2D, or transcription factor 2D complex. TF2D is the complex that actually binds to the Tata box or the initiator motif, and it does this using its TAF domain subunits. TF2D contains about 12 subunits called TAFs, which stands for TBP associated factors, and they bind to core promoter elements. Another important player in promoter localization is the mediator complex. It's a large or huge 2631 subunit complex that is thought to function as the bridge between RNA polymerase II and various transcription factors. So many of the transcription factors that you've heard of probably bind to one of the subunits of the mediator complex called MED1 through 31, MED1 through 31. For example, MED6 binds to estrogen and thyroid receptor transcription factors. MED12 binds to SOX9 and PGC1 alpha transcription factors. A general rule of thumb is that if you have a transcription factor binding to an enhancer element, it's probably achieving transcriptional activation by interacting with a subunit of the mediator complex. But the mediator complex itself is pretty poorly defined because really it's just a conglomerate of proteins that function as an adapter between transcription factors and RNA polymerase II. It's nothing more than that. Once TF2D and the mediator complex localize the RNA polymerase II holoenzyme to the core promoter, the DNA needs to be opened and made accessible. And this is achieved by the TF2H complex. TF2H is responsible for generating the transcription bubble. So I think of the H as meaning heat, as in to melt the DNA, because that's what TF2H does. It melts the DNA, although not literally with heat. I just think of the H as meaning heat. But anyways, there are many, many other players but in this entire process, but generally they have less interesting roles. Like TF2F, for example, increases processivity. TF2A aids in the binding of to the Tata box. But really the interesting proteins, at least in my opinion, are the mediator complex, bridging the hollow enzyme to transcription factors, TF2D, binding to core promoter elements, and TF2H, which is melting and unwinding the DNA to generate the transcriptional bubble. Once our complex is in place and ready to transcribe some RNA, it needs to go through a process called promoter escape. Because this whole time we've been talking and discussing how RNA polymerase localizes to promoters and all the different things that it binds. But if all these factors all bind to RNA polymerase 2, how does RNA polymerase escape them to move down the length of DNA and transcribe RNA? And this is the process of promoter escape. It's a complex mechanism, but to summarize what happens, I'll just say that a newly transcribed nascent RNA strand of only 14 or 15 base pairs, it actually protrudes out of the RNA polymerase. And this emerging RNA transcript alters the conformation of RNA polymerase 2 in such a way to free it from the promoter. And although probably technically incorrect, I personally envision this RNA strand kind of poking out of RNA polymerase, and maybe it physically pushes away from the mediator or the TF2D somehow. Although again, I'm not sure if that actually happens, but that's kind of what I envision, what I imagine. So the very short nascent RNA strand that emerges from RNA polymerase kind of destabilizes this entire initiation complex and instead favors the elongation RNA polymerase conformation. And then in the next slide, we're going to discuss the process of elongation. RNA elongation consists of two primary conformations, a stalled RNA polymerase 2 and a processive RNA polymerase 2. The stalling of RNA polymerase 2 is it's a very underappreciated phenomenon that regulates transcription. It's estimated that up to 20% of all RNA polymerases in your body right now are stalled and not moving. Negative regulators of RNA polymerase include, most importantly, the NELF and DCIF. NELF, which stands for negative elongation factor, does simply that. It inhibits elongation. The precise mechanism maybe isn't important, but DCIF accompanies NELF in a complex to promote transcriptional stalling. And just to illustrate how important this process is, I'll mention a study that found that the depletion of NELF increased the amount of HIV transcription by more than double. Researchers Henderson et al. Back in 2007 found that siRNA-mediated knockdown of NELF increased HIV transcription by more than double, about 2.5-fold, more HIV compared to controls. So this NELF complex is really important for the regulation of some genes. And NELF and DCIF are displaced by two main events: phosphorylation by PTEF beta or PTEF B, and also enhancer RNA. Enhancer RNA is a type of long non-coding RNA that has recently been shown to actually bind NELF, and it functions as kind of a decoy because this enhancer RNA it binds to NELF and it prevents it from inhibiting RNA polymerase 2 elongation. Right here you can see NELF binding the RNA that's coming out of the polymerase 2. Well, enhancer RNA can come in and it can act as a decoy to bind NELF and prevent it from associating with the elongation complex. Another important regulator of NELF is PTEF-beta. PTEF-beta stands for positive elongation factor B, and it's the name of a complex actually composed of CDK9 and one of many different cyclins, like cyclin T, for example. So PTEF-beta is technically a cyclin-dependent kinase, but it's a kinase that is critically important for RNA transcription, because without it, RNA polymerase cannot escape promoters, and it's the deletion of PTEF-beta actually, or its inhibition with a small molecule actually causes cell death. So PTEF-beta is really important, and it achieves transcriptional activation by phosphorylating NELF, causing NELF to dissociate. PTEF-beta also phosphorylates RNA polymerase's CTD, which stands for C-terminal domain. Its CDT, C-terminal domain of RNA polymerase II, is commonly known as RNA polymerase CDD tail. This CTD tail is basically the master regulatory domain, of RNA polymerase II, and it can be phosphorylated at a couple different really important sites. CTD phosphorylation at serine 2 by the PTEF complex greatly enhances its processivity. And although I'm not sure of the mechanism, it's important. Regardless, the idea here is that PTEF-beta recruitment to a stalled RNA polymerase is critical for processive elongation of the RNA polymerase complex. And the recruitment of PTEF-beta itself is thought to be mediated by the 3' untranslated sequence in the emerging RNA transcript. And the final step in transcription is termination. And I don't have a slide for termination since I couldn't actually seem to find a single agreed upon model. But what I've gathered is that there exists a termination sequence that is ill-defined, but it binds to an exonuclease called XRN2. XRN2 is an exonuclease that chews up the 5' UTR so quickly that it actually pulls the RNA out of the RNA polymerase. This is known as the torpedo model, because the XRN2 exonuclease is like a torpedo that runs right up into the RNA polymerase and basically pulls the RNA transcript out of the polymerase. Another model for RNA polymerase termination suggests that a short palindromic sequence at the end of the transcript causes a short hairpin loop to form inside of the polymerase. This causes the RNA polymerase to stall and dislodge itself. But the summary of this slide is that NELF and DCIF are really important negative elongation factors that can be phosphorylated by the PTEF-beta complex, causing NELF to dissociate. And termination is likely done by the XRN2 exonuclease. During transcription, the emerging RNA transcript is very actively processed by various enzymes. There are 3 general things happening to the RNA transcript during elongation. We have 5' capping, splicing, and immediately following the transcript's release, we have polyadenylation. The 5' cap, which emerges first, is recognized by the mRNA capping enzyme complex, which consists of 3 different proteins. The capping enzyme complex itself is recruited to the RNA polymerase by CTD tail phosphorylations. In the next couple of slides, we're going to see just how important the 5' cap is for export and translation. The mRNA funneled out of the RNA polymerase is also bound by various SR proteins. SR proteins are serine-arginine enriched proteins that use the positive charges of serine and arginine to bind the negatively charged phosphate backbone of RNA, and they function to delineate exons and introns for splicing. We'll discuss that a little bit more in the next slide. And the S and the R stand for serine and arginine as well. The emergence of the 3' UTR opens a binding site for a protein called CPSF. CPSF binds to the mRNA and it cleaves it off, producing its own 3' end. CPSF then recruits a polyadenylate polymerase, or simply poly-A polymerase. Poly-A polymerase simply catalyzes the nonspecific poly-A tail that is recognized by poly-A binding proteins, which are important for many of the downstream processing, transport, and translation of the mRNA. And lastly, we have a 5' UTR. 5' UTR, which we'll see in the translation slide, is very important for ribosome binding. I want to say a quick word about splicing. Although it's obviously a very interesting and important topic, I don't want to spend too much time on it because it's honestly, it's confusing, it's complex, and ultimately I've noticed after reading about the spliceosome, I end up not even understanding what I just read. So I feel like splicing is also extremely transcript specific. So trying to discuss generalities about splicing is a very limited topic. But there are some things more or less universal about splicing. Exons and introns are bound by the SR proteins, a family of about 12 different proteins that regulate splicing by either promoting or inhibiting splicing, a process called splice site definition. Generally, SR proteins bind to exons to prevent their cleavage, but some SR proteins can bind to introns to promote their removal. And SR proteins do this by interacting with splicing machinery to guide the spliceosome to introns. SR proteins are also fairly heavily regulated by phosphorylation, which can do things like induce nuclear export. So imagine an SR protein that normally binds to exon 3 or something of some gene. If that XR protein is exported from the nucleus due to a well-placed phosphorylation, then perhaps that exon 3 of whatever gene that was will now be spliced out, giving rise to an alternative splicing event. And I'm not going to go down that rabbit hole, but just be aware that SR proteins are dynamically shuttled and they're regulated by phosphorylation. And that's one important mechanism of coordinating alternative splicing is by modulating SR protein dynamics. The spliceosome assembles and cleaves the introns through a complex dance of 30 or 40 proteins and some RNA called snRNA. snRNA, or small nuclear RNA, they are utilized for their ability to recognize certain sequences via complementary base pairing. And this aids in the proper splicing of mRNA. I think the summary is that SR proteins define exons and introns, and they can be regulated to mediate alternative splicing. And RNA protein complexes that make up the spliceosome remove introns. And down here in panel B, we see the recognition of downstream sequence elements by CPSF. CPSF then cleaves the transcript and recruits poly-A polymerase. If we zoom out a little bit and look at where all this mRNA processing is actually occurring, we actually see areas of active transcription. These green speckles are called nuclear speckles, and in the image to the left, they are actually marked by antibodies towards U2. U2 is a critical component of the spliceosome. So it would be a misconception to think of the nucleus as an amorphous RNA transcribing organelle, because there's actually very distinct subcompartments for active transcription, and these are called the nuclear speckles. And actually transcriptionally silent areas or inactive regions of the DNA are wrapped up in chromatin called heterochromatin, seen here on the right. Heterochromatin accumulates typically near the edges of the nucleus, where they're packed very tight by lamin proteins and other nuclear membrane-bound proteins. Once our mRNA has been capped, spliced, and polyadenylated, it needs to be exported from the nucleus. And this process of nuclear export is increasingly being recognized as yet another mechanism of gene expression regulation. Selective export of mRNA to the cytoplasm for translation can actually be modulated by kinases and other factors in order to remove or promote gene expression. Formation of an mRNA export competent complex called TREX, T-R-E-X, or TREX2, is responsible for freeing mRNA from nuclear speckles. Many components of the TREX complex bind to splicing factors like SR proteins, thus localizing the mRNA export complex to spliced mRNA that's ready to be exported. A couple components of the TREX complex bind directly to mRNA, like the ALY protein, ALY protein. However, the most important mRNA binding protein is the NXF1 protein. NXF1 is simply an mRNA binding protein that can only bind to mRNA that is in complex with the TREX complex. So TREX binds to mRNA during or after splicing, removes it from the nuclear speckles, frees it from the nuclear speckles, and once TREX has assembled itself on the mRNA, the NXF1 protein is then able to bind the mRNA. The TREX complex kind of unlocks the NXF1 protein and turns it into an open conformation that can now bind RNA. Once the mRNA TREX NXF1 protein complex has constructed around the mRNA, it actually begins diffusing around the nucleoplasm until it binds to a nuclear pore. This is considered the rate-limiting step and actually takes several minutes of random Brownian motion until it encounters and docks with a nuclear pore. The docking of the TREX NXF1 mRNA complex with a nuclear pore occurs through the interaction between NXF1 and the nuclear pore protein called nucleoporin. Nucleoporin. The nuclear pore is actually made impermeable by protein filaments composed of hydrophobic phenylalanine and glycine repeats, or FG repeats. F for phenylalanine, G for glycine. These FG repeat filaments on nucleoporins compose a kind of sieve that prevents non-specific diffusion, right? The NXF1 protein bound to mature mRNA is recruited to the nuclear pore FG repeats, and this actually leaves behind these the very large TREX complex. Once in the nuclear pore, the NFX mRNA complex is pulled through by the action of the DPB5 protein. DBP5 is an RNA-binding ATP-dependent helicase. DPB5 also dissociates from mRNA, dissociates the mRNA from NXF1 once it's in the cytosol. Okay, so to summarize, we have SR proteins, the 5' cap, and various splicing proteins recruit the TREX complex. The TREX complex helps mRNA escape the nuclear speckles and also allows the NXF1 protein to bind to mRNA. NXF1 bound mRNA diffuses through the cytoplasm, and eventually it encounters a hydrophobic FG repeat inside the nuclear pore. Inside the nuclear pore, these NXF1 mRNA complexes are recognized by the DBP5 helicase that pulls the mRNA through the pore and basically releases the mRNA into the cytoplasm. This slide provides a nice summary of nuclear export. So we have our TREX complex. It binds to the mRNA by the recognition of several factors, including SR proteins and the 5' guanosine cap. The TREX complex unlocks NXF1, allowing it to bind mRNA. NXF1 coats the mRNA, and it diffuses until it binds to the FG repeats inside of the nucleoporin. DBP5 will then reach in and grab NXF1 and pull the mRNA out of the nuclear pore into the cytoplasm. Before moving on to translation, I need to mention the CRM1 protein. CRM1, also known as exportin 1, is the protein responsible for tagging and marking proteins for nuclear export. And since many of the proteins that bind to mRNA also contain a nuclear export signal, many mRNA are actually exported indirectly by CRM1. ELAV1, also known as HUR, is an example of an RNA binding protein that can basically function as an adapter to CRM1 for nuclear export. And besides mRNA, tRNA and microRNA tRNA and microRNA are primarily exported by EXPO5. EXPO5 is a homolog of CRM1 that does pretty much the same thing, except EXPO5 can bind directly to the microRNA or the tRNA and be exported. And we'll discuss EXPO5 a little bit more when we talk about microRNA. After splicing, capping, and polyadenylation, mRNA can also be methylated at adenine residues, in addition to a bunch of other modifications that I don't really have time to talk about. But this methylation is called M6A, and it's by far the most studied post-translational RNA modification. M6A is catalyzed by a complex of MET-ELF3 and MET-ELF4, and is actually reversible by the demethylase FTO that you see below it. M6A alone doesn't actually do anything, but M6A can be bound by RNA binding proteins that are specific for methylated adenines. These RNA binding proteins that are specific for methylated adenine are called M6A readers. And this includes the YTHDF1 through 4 family of proteins, who are in desperate need of a new name. YTHDF proteins and a couple of other proteins which have been shown to bind M6A, they mediate the very diverse effects of these methylations. M6A has been linked to increased mRNA export, it's been linked to mRNA destabilization, and enhanced translation, likely depending on the specific location of the methylation. M6A methylations at the 3' UTR appear to promote degradation in P-bodies, generally, which we'll discuss in a bit what a P-body is. But methylations near the 5' guanosine cap in the 5' UTR, these ones appear to promote translation by recruiting translational machinery. And at the level of the whole organism, deregulation of the MET-EL3 methylase has been implicated in cancers. Certain cancers have been shown to be dependent on MET-EL3 for survival. In addition, the single most highly correlated mutation linked to obesity is, strangely, in the FTO gene, which I just found bizarre. So for reasons that are not understood, the inability of FTO to demethylate M6A residues increases the risk of obesity. But exactly how this genotype correlates into a phenotype is, I don't understand that, but I do find that really interesting. So although this modification is very understudied, and research into it is basically in its infancy, it's increasingly being recognized for its role in cancer and also metabolic disorders, and maybe even more. Once the mRNA has been spliced, modified, and exported, it's now suitable for translation. In this graphic, we have basically the bare minimum machinery needed for translation initiation. And this is the pre-initiation complex. The process kicks off with our mRNA 5' cap and polyadenylation tail that nucleate the complex. The 5' cap recruits EIF4E, and the 3' polyadenylation tail recruits EIF4G. The recruitment of EIF4E is actually considered the rate-limiting step in translation, and it's also the most highly regulated step. EIF4E binds specifically to the methylguanosine cap of the mRNA. And the disease relevance of this protein is kind of fascinating as well, because mutations in EIF4E have been linked to autism, and mice that overexpress EIF4E are prone to autistic-like behavior and also cancer. And this is partly due to translational dysregulation caused by mutations in EIF4E. A protein, cleverly named EIF4E binding protein, abbreviated 4EBP, can bind and inhibit EIF4E, preventing it from binding to the methylguanosine cap. And this prevents translation. This 4EBP mediated inhibition of translation is actually a super common pathway in cells, whereby stress signaling, viral infections, protein misfolding, and many other stressors, they often converge on the activation of 4EBP to inhibit translation. So anytime you have stressed out cells that need to conserve ATP, they often do this by inhibiting translation using 4EBP. Our poly A tail is bound by poly A binding proteins, and these poly A binding proteins help to nucleate the pre-initiation complex by recruiting EIF4G. EIF4G is a kind of scaffolding protein that circularizes the mRNA and helps to promote the polyribosome structure. So the polyribosome, which means multiple ribosomes, right? Poly meaning many. You can have upwards of 20 or 30 ribosomes that bind on a single mRNA, and they generate many proteins simultaneously. And the presence of EIF4G is really important to that process because it circularizes the mRNA. So when one ribosome finishes translating, it can pretty quickly jump right back to the beginning, the 5' cap, and then reinitiate translation. And so EIF4G is really important for the rate of translation, and it also helps to stabilize the pre-initiation complex. The EIF4E, A, H, and G proteins, all of these, they constitute the EIF or EIF4F holoenzyme. So EIF4F is the complete pre-initiation complex. And what it does is it recruits the small ribosomal subunit and a methionine-charged tRNA. This pre-initiation complex, which remember is located at the 5' untranslated region, will begin moving down the mRNA and basically will be looking for the start codon, the AUG. This process is called ribosome scanning, where the EIF4F complex and the small ribosomal subunit and also the methionine-charged tRNA will scan the 5' UTR for the start codon. It'll start looking for the region to begin translation, which, as you can see in this graphic, is right over here. And the complex is able to recognize this start site because the complementary base pairing between the mRNA and the methionine tRNA. Base pairing between these two RNAs causes a conformational shift that activates GTP hydrolysis by EIF2. And this somehow destabilizes the pre-initiation complex, and it allows the large ribosomal subunit to bind. So I don't want to get into specifics because it's really complicated, and the specifics probably aren't too important, but basically what happens is the small ribosomal subunit in conjunction with EIF4F will scan the 5' UTR until it finds this AUG. The methionine tRNA will base pair with the AUG, and this base pairing is what allows the large ribosomal subunit to come in and bind. So the ribosome is actually basically constructed on the spot at the start codon of the mRNA. One really interesting thing about this 5' UTR is that it's actually, in fact, very dynamic. Since the 5' UTR is the site of translation initiation, and it's also rate limiting, it can thus serve as a critical site of translation regulation. One frequently studied example is the so-called IRE sequence, IRE, for iron regulatory element. The IRE is a 5' sequence that forms a stem loop structure after transcription that can bind to specific mRNA binding proteins called IRP1 or also IRP2. In this graphic, they're called IRBPs, IRE binding proteins. These IRPs will recognize and bind the stem loop, and they simply prevent translation initiation. As you can see here, there's no translation initiation. However, when iron accumulates in the cell, the iron can actually bind these IRP1 and 2 proteins at an iron-sulfur group, and this causes them to release the stem loop structures and allow translation. So the idea is that the 5' UTRs are not only the site of translation initiation, the rate limiting step in translation, but can also be precisely regulated by RNA binding proteins due to unique secondary structures that can form within the 5' UTRs. Since we're discussing RNA and translation, I think it's worth mentioning tRNA. tRNA is the bridge between RNA and protein. They are the mechanism through which specific codons code for specific amino acids. And there exists a tRNA for each codon. There's about 35 in total. And each tRNA contains a unique anticodon that recognizes and base pairs with particular mRNA codons. And this is the mechanism by which tRNA recognizes specific mRNA codons. However, before being sent to read the mRNA for the ribosome, the tRNA needs to be charged with an amino acid. So the tRNA is covalently attached to a specific amino acid, and this occurs at the 3' end of the tRNA. Right here, you can see it's actually charged, it's attached to an amino acid. I'm not going to get into the specifics, but basically each tRNA has a partner amino acid tRNA synthetase, an enzyme that will charge it with an amino acid, so that when the tRNA recognizes its anticodon in the ribosome, it can transfer that amino acid to a growing peptide. So for example, the tRNA that recognizes glutamine will be charged with by the glutamine binding tRNA synthetase. The tRNA that recognizes the leucine codon will be charged with the leucine amino acid by the leucine tRNA synthetase. And the system is it gets a lot more complex when you consider things like wobble base pairing, inosine, or other things. So trying to determine exactly how the amino acyl synthase actually binds to specific amino acids is another complex issue. But we're not going to get into all that. The basics are just that. Now we're going to discuss how mRNA is degraded. We've gone through how it's transcribed, how it's modified, how it's translated. Now let's discuss how it decays. There are 4 general routes of mRNA decay. We have deadenylation, nonsense-mediated decay, non-stop mediated decay, and no-go decay. In deadenylation, what happens is that an mRNA transcript, its 3' poly A tail is competitively bound by various deadenylation enzymes that are constantly competing with poly A binding against poly A binding proteins. So the PABP is the common poly A binding protein, and PABP binds and stabilizes the transcript by binding the 3' tail. But if it's displaced for whatever reason, It could be phosphorylation. It could be another protein complex. When PABP is displaced, deadenylation complexes can bind. And 3 identified mRNA deadenylation complexes that have been identified are the PAN23, the CCR4 NOT, and the PARN, PARN complex. The CCR4 NOT complex seems to be the most popular, or at least most well studied of these deadenylation enzymes. Generally, CCR4 NOT is a kind of like a passive deadenylation. It's kind of like a clock. It sets a time limit to mRNA. So the longer the mRNA has been around, the more its poly-A tail gets chewed up by the CCR4 NOT enzyme. And eventually, when the poly-A tail has been depleted, it gets recognized by specific or non-specific RNases in what's called the exosome complex. There's actually lots of different RNases in the cytosol that do this, and it has huge implications for RNA recovery during experiments. And their biological function is to protect against viruses and also degrade mRNA that has lost its poly-A tail due to these deadenylation enzymes like CCR4 NOT. Deadenylation can also expose or can also trigger decapping by the decapping enzymes DCP1 and DCP2. And once a mRNA has been decapped, it exposes the 5' UTR to the XRN1 RNase. So next time you go to harvest tissue for an experiment, just realize that every second that passes, this pesky CCR4 NOT complex is chewing up poly-A tails and it's exposing your precious RNA to internal RNases in the sample. So this system right here represents a more passive mode of mRNA decay. It just kind of happens in the background. It's basically a clock for the mRNA. It gives the mRNA a time limit. In the next few slides, we're going to discuss a more active and regulated mechanism of RNA decay. There are 3 common examples of mRNA that you do not want to get made. So not all mRNA will make a suitable protein, right? mRNA that has a premature stop codon, mRNA that for some reason causes a ribosome to get stuck, or mRNA that doesn't contain a stop codon. These are all mRNA species that you do not want in the cytosol. And the cell has actually evolved clever mechanisms of recognizing these defective mRNA and disposing of them in what's called the exosome. The exosome is just a collection of RNases that degrade mRNA. And we'll discuss a little bit more on the next slide. In nonsense-mediated decay, we have a premature termination codon. I think of this as a transcript makes absolutely no sense because it only codes for half of the transcript. It makes no sense. So it's a nonsense-mediated decay. And it's orchestrated by these UPF proteins, 1 through 3. There's actually 3 of them. There's a bunch more SMG proteins as well. But these UPF proteins recognize translational termination that occurs before all exon junction proteins have been displaced. So it's pretty ingenious. What these UPF proteins are doing is that they are looking for ribosomes that terminate without displacing all of the exon binding proteins, indicating a transcript that has only been half translated, presumably because there's an internal stop codon. So it's a fairly complex process, but basically, if the components of the ribosome dissociate while UPF1 is still bound to an exon junction protein, it activates UPF1 and it initiates decapping and mRNA decay. In non-stop decay, the original mRNA transcript lacks a stop codon, so it's non-stop translation. And when the ribosome reaches the end of an mRNA without finding a stop codon, it leaves the ribosome in a very awkward position where its entry and its peptidase domains are basically left open. So remember that ribosome has the E and the P and the A tRNA binding sites. I didn't cover that, but anyways, the open site in the peptidase in the entry domain without any codon below it serves as a binding site for this SCI7 protein. The SCI7 protein binds to the empty entry site at the ribosome because it kind of resembles a tRNA, and then it can somehow recruit the exosome and all the necessary RNases. And lastly, we have no-go decay. No-go occurs when the ribosome stalls. It can no longer go because perhaps there's a codon that doesn't match any tRNA, or perhaps there's a secondary loop structure that impedes elongation. And this is the most recently discovered type of RNA surveillance, but it involves the DOM34, also called HBS1 complex, that like SCI7, it binds to empty entry sites at the ribosome and it initiates degradation. So just to summarize, if the transcript has a premature stop codon, the UPF protein will interact with ribosome termination factors and be activated and recruit decapping enzymes. If the ribosome runs all the way to the end of an mRNA without finding a stop codon, the empty entry site on the mRNA can be recognized by SCI7, which then recruits the exosome. In no-go decay, something similar happens. A stalled ribosome's entry site is recognized by DOM34, and this recruits RNases. When a defective mRNA has been identified by any of the mRNA surveillance mechanisms, it is degraded not usually on the spot, but rather it's first transported to a P body. The P stands for processing body. P bodies are kind of pseudo-organelles that don't contain a membrane or anything, but they instead are these sites of decapping enzymes and exonucleases like XRN1. These mRNA decay enzymes are, so they're not really cytoplasmic, but rather they hang out in these P bodies. Thus, a defective mRNA needs to be transported there first in order to be degraded. And I personally prefer the term P bodies instead of exosomes. These are also called exosomes because it's a complex of all these different processing mRNA processing proteins. But the exosome is kind of a strange name because it's also the name of an extracellular vesicle, and exosome doesn't really make any sense to me. So sites of mRNA degradation are in P bodies, and sometimes they can also be nucleated into very large stress granules through the action of this G3BP1 protein and TIA1, TIA, is supposed to be 1. But the last type of mRNA regulation occurs through the utilization of microRNA and the RISC complex. RISC stands for RNA-induced silencing complex. This field of mRNA regulation has definitely generated the most excitement, and rightfully so, because it has basically revolutionized molecular biology. And we'll discuss how that works. So at the 3' UTR of our mRNA, there often exists what's called a seed region that can be recognized by an RNA-dependent endonuclease, which is the RISC complex. RISC is technically nothing more than an RNA-dependent endonuclease, but it requires complementary base pairing in order to cleave its target mRNA. And one of the most important facets of the RISC complex is where that RNA comes from. RISC is loaded with RNA, and that RNA determines the endonuclease's specificity. So this RNA that's loaded into RISC can come from 3 main sources: viruses, the host genome, where it's called microRNA, and also it can come from humans who inject it or electroporate it. It's called siRNA at that point. So let's talk about how the cell generates its own microRNA that is loaded into the RISC complex. In order for this microRNA to ultimately go on to induce mRNA cleavage, it has to be complementary to the 3' UTR of its target mRNA transcript. If the microRNA needs to be complementary to the mRNA, then where do you think the microRNA comes from? It actually usually it comes from the same gene. It's simply transcribed in the reverse direction by RNA polymerase. Sometimes the microRNA is found within introns of its target gene, allowing the microRNA to be generated at the same time the mRNA is generated. It's really quite ingenious. Regardless of where the microRNA comes from, once it gets transcribed, it folds into a very important stem loop structure. This secondary structure is required in order for the microRNA to be processed. So pre-microRNA stem loop structure is recognized and bound by the Drosha DGCR8 complex. DGCR8 is what actually binds the pre-microRNA, and Drosha is the actual endonuclease that cleaves all these single-stranded regions of the microRNA out. So Drosha basically cleaves everything out but keeps the stem loop structure of the microRNA. But critically, it leaves a very small 2 base pair overhang. It leaves a 2 base pair overhang, which is important for, you can kind of see it in the graphic. This is a little 2 base pair overhang that's important for the recognition down the road. So this small 70 base pair stem loop structure is picked up by exportin 5, also called expo5, and it's transported out of the nucleus. Once in the cytoplasm, the stem loop is recognized by another endonuclease called Dicer. Specifically, the PAZ domain of Dicer recognizes that 2' or that 2 nucleotide 3' overhang that is left from Drosha. And Dicer cleaves off the stem loop, leaving just a short 22 base pair double-stranded RNA. Double-stranded RNA, including this short 22 base pair strand, now called microRNA, are substrates for an RNA binding complex, our RISC complex. RISC binds to double-stranded RNA, whether it's from a virus, the genome, or if a human injects it. We will see a more complete layout of RISC in the next slide, but it's fundamentally composed of this AGO2 protein, AGO2. Its full name is argonaute. And AGO2 will bind double-stranded RNA and selectively digest one of the strands using its PIWI domain, P-I-W-I, PIWI. So AGO2's PIWI domain selectively degrades one of the strands. The strand that gets degraded is called the passenger strand. It's not needed, while the strand that will go on to base pair with target mRNA is called the guide RNA or gRNA. So the RISC enzyme is essentially AGO2 with a single-stranded guide RNA that will now roam the cytoplasm, testing RNA for complementary base pairing. And if it recognizes a complementary strand, that other strand often gets degraded, or at least translation is inhibited. So just to summarize, microRNA gets translated. It folds into a stem loop structure. It gets cleaved by Drosha, exported by expo5, cleaved again by Dicer, and then that double-stranded RNA is picked up by AGO2, which degrades the passenger strand, keeps the guide RNA, and will begin looking for complementary base pairs to degrade. So what exactly does the RISC complex do once it finds complementary RNA? The RISC complex is composed of the catalytic endonuclease AGO2 and also this regulatory protein called GW182. GW182 is a kind of scaffolding protein that bridges the RISC complex to other mRNA degrading complexes. When AGO2 finds a complementary strand, it can, in addition to just simply cleaving the strand, it can also activate GW182 to recruit DCP1 and 2, the decapping enzymes. GW182 can also recruit the CCR4 NOT complex to catalyze deadenylation. So although AGO2 is itself an endonuclease, it alone doesn't appear to be very effective in digesting complementary mRNA, because the knockout of GW182 actually significantly impairs microRNA-mediated gene silencing, suggesting that the recruitment of decapping and deadenylation enzymes is actually a pretty important process for RISC gene silencing. You can also see the PAN3 and PAN2 complexes for deadenylation as well. So RISC uses its guide RNA to locate complementary base pairs, which are found at the 3' UTR, by the way. Notice this is a 3' UTR. And once it binds to the mRNA, it recruits decapping enzymes like DCP1 and 2, recruits deadenylating enzymes like CCR4 NOT, and also, if the sequences are perfectly matched, AGO2 can actually cleave the mRNA itself. And this is, again, this is occurring in P bodies, I should mention. I wanted to provide some examples of characterized microRNA. And although I would like to go into some detail, there's too many of them, and their precise functions have not really been worked out. But as you can see, there are many microRNAs confirmed to be involved in regulating toxic proteins like amyloid precursor protein or Huntington, which is over here. The sheer size of the human genome and the short 20 base pair length of a microRNA means that bioinformatic approaches to microRNA predictions are very dubious, and they need to be validated. For example, I used microRNA database to check out a couple of genes, and based on sequence similarity alone, the Huntington gene, which is what I checked out, is predicted to be targeted by 156 microRNAs. The amyloid precursor protein is predicted to be targeted by 33 microRNAs. P53 predicted to be targeted by 51 microRNAs. And what's strange is that the top predictions, with the best sequence similarity, are completely unvalidated. For example, the microRNA 6884 has a nearly 100% sequence similarity to 4 different locations in the 3' UTR of P53. But despite that, multiple high-throughput deep sequencing methods have failed to find this transcript, this microRNA, and even though it's a perfect match for P53, but apparently cells are confusing and they work in mysterious ways, and this microRNA is no longer functional, doesn't work. So even though it's perfectly situated to regulate P53, it doesn't appear to be actually occurring in the real world cell. So in other words, it's probably not transcribed, and if it is, it's not processed correctly. But anyways, I thought just knowing how the general system works is pretty valuable, but trying to use specific examples is hard to give a lecture on. For my last slide, I wanted to provide a quick shout out to another type of RNA that I was originally going to include some slides on this, but I decided not to. And this is long non-coding RNA, or simply called lncRNA, lncRNA. This type of RNA does just about everything you could imagine it might do. So imagine for a second, imagine some RNA in the nucleus, and try to picture everything an RNA could do with its ability to base pair with specific sequences. And lncRNA probably does all of it. LncRNA is non-coding RNA that uses base pairing to bind and regulate many other things. For example, lncRNA can function as a decoy. Perhaps there's an RNA binding protein that normally binds mRNA. Well, maybe there's a species of lncRNA that has evolved to mimic that target mRNA, and maybe it functions as a decoy. It might do this to, hold it in the nucleus. Or perhaps there's some lncRNA that functions to bind microRNA to hold it in the nucleus like a sponge. And perhaps there's lncRNA that associates with a protein, and maybe it gives the protein highly sequence specific binding affinity in order to regulate splicing or degradation or export. Maybe the lncRNA binds to histone modifier proteins to direct it to specific DNA sequences. All these general phenomenons have been validated to occur in the human genome. So I'm not going to go into more detail, but I'll summarize lncRNA as doing just about everything an RNA molecule could reasonably do in the nucleus. I envision lncRNA as a type of protein. I envision it like a protein that basically has the ability to bind RNA or DNA in sequence specific manners, which, if you think about it, that could be extremely useful for a cell, and it really is a useful ability. Anyways, that's all I have for mRNA and microRNA. I hope you guys enjoy the presentation. And if you have any questions or future lecture requests, please comment and let me know. And thanks for watching.